Good afternoon. My name is James White. I'm the CEO and owner of KBMO Diagnostics. And we're here today. We've been kindly invited to talk a little bit about our COVID-19 rapid test, which we've now enabled to be run at home. So we want to update everyone on the progress that we've made. And I think it's worth mentioning, we've had this in place for probably about a week, 10 days now, but we're trying to be very cautious to make sure that we wanted to release it the correct way. So let me start by going through a little bit of background on the company, and then we can talk about our operations and how we're set up now to hopefully launch the test early this week. So the company was founded by Dr. Brent Dorval, who was an MIT, and he also discovered the first rapid HIV diagnostics. And I'll talk a little bit more about his background, and you guys will also have the pleasure and honor, frankly, of listening to him talk about COVID-19. And given his experience with HIV, it's very pertinent to kind of get an infectious disease specialist of his type on to give some background on not only COVID-19, but the test that we've chosen and some of the reasons why we've chosen it. Ordinarily, we're a company that focuses very much on food inflammation testing. So that's obviously very important when you think of immune health, to look at making sure that via your diet, you can reduce your levels of inflammation. So we consider that an important test, but we wanted to make sure that we had something a bit more relevant again by looking at a COVID-19 test. So ordinarily, as I say, we're a full service diagnostic company. We manufacture tests in our clear high complexity lab. We do have a ISO 13485, and it's also an FDA registered facility. We've got just over 20,000 square foot, and we offer our tests all around the world via a combination of test types, either blood draw or finger stick. And with this COVID-19 test, we'll talk about a little bit about what we're currently doing with the serum offering that we'll have this week. And then hopefully, as we validate the blood spot offering, how that could also be helpful as an in-home test, as well as an offering for physicians who are working from a telemedicine standpoint, and equally how some of the things we've come up with on the serum test should be able to be helpful there as well. So again, as I mentioned, the management team, I've been involved in diagnostics for a number of years and been very successful in terms of taking products through the FDA, launching those, as well as acquiring products in. Way more importantly though, Dr. Dorval, who'll be speaking next, he was the founder of the company. As I mentioned before, he invented the HIV diagnostics, the first rapid one in 60 seconds, and was also a key advisor to the WHO Vaccines and Diagnostics Committee in Geneva. So I think it's very relevant and pertinent to have him talk to you. And I guess in the words of the head of the WHO, his advice to every country now is to test, test, test. So to get Brent's insight into that, I think hopefully you'll find that very helpful. Anthony Recupero, he's our Chief Operations Officer and has been instrumental on growing our facility to, and you'll see how many tests we've run a year on the next slide. And lastly, but certainly not least, is Dr. Jahi, who has much experience in both the USA and China, PhD, and also board certified lab director for 3 labs here in the US, and has been very helpful as we've been sourcing the right type of test and the right type of technologies out of Asia. So again, the lab team, there's a picture of the lab techs outside our facility. We're just outside Boston in Hopedale, Massachusetts, and they ran a staggering 2.5 million assays last year. So they're well capable and very much well set up to be running this COVID-19 assay. We've chosen the one that we have, and Dr. Dorval will go into more detail on the test itself, but mainly because the test that we've gone with is the first test, the first colloidal gold antibody test that was approved by the Chinese FDA, and it's also run over 2 million tests out there. So again, as you see a number of tests come through the FDA at the moment, I think it's great news that they're approving lots of tests. Back to my comment from the head of the WHO of test, test, test. We won't be able to do all the tests for the 350 million Americans out there, but we've gone and chosen and partnered with the group that's done over 2 million tests. So again, I think it's an important number to kind of reiterate to you that this is a battle-hardened test. It's being used in Wuhan, of all places in China, which again, I think most people are now familiar with. So with that, I'll hand you over to Dr. Dorval, who can talk a little bit about the technology and why we've chosen this test.
Thank you, James. It's a pleasure to speak to everybody this afternoon. This is Dr. Brent Dorval. I'm the Chief Scientific Officer of KBMO Diagnostics. I'd just like to go over a few important features of the COVID-19 virus and its relevance to the assay that's been developed and that we're using at the KBMO facility. There are four main structural proteins for this particular virus. It's a circular virus. It's about 150 nanometers in diameter, and there are four main proteins, as I just said. The spike glycoprotein, there's the RNA with the N protein associated. That's a helical nucleocapsid. You have an envelope, and then you have the hemagglutinin esterase protein, which is very, very similar in many ways to the influenza hemagglutinin esterase found on that particular virus. These are the main structural proteins that we deal with this virus, and the one that is applicable to the test that we use is the N protein. And the reason we use this is that it comes up very early during infection, and I'll go over that in a little more detail in a few minutes. There are a few main diagnostic methods that are currently used. Some are preferred and some are kind of archaic. The first one is isolation in cell culture. That's an archaic method. It involves growing live virus. Although it works very well, it's tricky, it's dangerous, and you need BL3 certification, which is a very, very high level of containment for this particular virus. Nucleic acid detection is an early detection method, and that is really the gold standard today. The next one is antigen detection, and that can be done by a method called Western blot. That will detect these particular viral proteins on a gel matrix, and then you can identify them with specific antibodies. It's a good method, used infrequently, and really only in specialty labs. The method that we're using, and we'll speak about today, is IgM and IgG detection. These antibodies are produced, especially IgM, early in infection, and it is against the nuclear protein in particular. IgG comes on a little bit later, and it's also against the nuclear protein, and it's a very, very sensitive method to detect this virus. And with this assay, also very, very quick. We can do this within 15 minutes from sample to result. The next slide, we can just go over briefly the kinetics. That means how does an infection occur, and then when does antibody become detectable? As you can see in the top slide, you can look at IgG antibody expression. You can see the highest line there, the N protein, comes up around 3 to 5 days and shows an incredibly strong response. The middle line shows the M protein, which is a membrane protein. That's a very weak response, and it's, although prolonged, it's very, very weak, and detection is low. And as you can see, the spike protein, which is that large protein in the membrane, it is slow to rise, but later on comes on a little bit stronger. Now, if you look at the next slide, which is the IgM expression profile, as you can see, much like the top slide, the N protein comes up highest and strongest, although less than for IgG, and it comes up a little bit, it seems to come up a little bit later in the strongest, for the strongest response. Next is the M protein, which is at the very bottom, very weak response, and in the middle is the S protein. So on our test, what we do is we use the nuclear antigen to capture IgM and IgG from the human serum sample, and then we measure it with colloidal gold to show that if you're positive, you get a strong response and a nice bright red line for either M or G. In the next slide, this is just a depiction, a kind of a pictogram of the actual test itself. As you can see, it's a simple plastic device. And what you do is you add the sample in the port labeled S. On one side is IgM, on the left, and on the right side is IgG. So you simply add 10 microliters of serum, or you can even use whole blood. Right now, we're using serum as the preferential standard. So what happens is the sample contacts the colloidal gold that's coated with the nuclear antigen. When that happens, the serum antibodies bind to the nuclear antigen, migrate past the test line, that's T. And if there is an IgM or an IgG antibody against nuclear antigen, it's captured on the test line, and that will show up as a red line. In this particular sample that you see, you can see on the right side, on the left side, the person is IgM negative, and on the right side, you can see two lines, the test line in particular, that is IgG positive. In any event, if the patient were negative, that means there were no test line visible. The control line C would light up, and that would tell you that the test was performed correctly. So this test has been used on several million patients in China, and James partnered up with that company to bring this technology heretofore into the United States and into the KBMO diagnostic laboratory. In the next slide, what you can see again is just a simplistic pictogram of the test. So if you're positive for either IgG or IgM, you'll see two lines. If you're negative, you see one line. Sometimes, but very infrequently, you may get one line and the control doesn't show up, the line by the C. That would be an invalid test. And if no line showed up, of course, that would be invalid. So you always want to see at least the positive control show up. That tells you that the test was run properly. So it's a simple test. It's really hard to make a mistake, but anything's possible, and occasionally you would get an invalid result, and in that case, you simply repeat the sample. In the next slide, the product is based on, as you could see, it gives you a clear indication of whether the patient is positive or negative, and when they use this at the Chinese CDC, it was conducted at 5 different institutions, and a total of 447 cases were used. And using this kit, 110 out of the 126 confirmed cases are positive, and it shows a sensitivity of 87.3%. 62 cases of the clinically excluded cases are totally negative and showing a specificity of 100%. What's interesting is that what you'll see is if sometimes the nucleic acid test is used, they call it an RT-PCR, a real-time PCR, sometimes you can detect the virus very, very early in infection, maybe within a day or two, so that you can be PCR positive, but you can be antibody negative. So sometimes a clinically positive person would be just not symptomatic, but just coming into the infection, and then the antibody would develop later, so they would be PCR positive, but IgG negative.
Thank you, Brent. That was a great description of the technology and the science behind it. Again, sometimes with all the amount of misinformation going out, it's good to hear it from a WHO expert like Brent. So again, what I thought I'd try and do is highlight that, as I mentioned earlier, it's great that the FDA is hurrying through some tests now that just fresh onto the market. What we wanted to do is, again, partner with a group that had significant experience, and the list of locations here all around China, as well as more specifically in Wuhan. It says 50,000 at the bottom, but actually that is 2 million tests, because there's 40 kits per, or 40 tests per kit. So again, this has been extensively used, and that's really why we were keen to work with this test, because again, at the end of the day, this is a major pandemic we're dealing with. We're dealing with a serious infectious disease, and we want to make sure we're working with a test that has been truly road tested, and that's not just a few in a lab. We want to work with one that has got significant backing to it. One of the other good things about this test as well, that our partner has got the capability of manufacturing up to 200,000 tests a day. So again, if you look at how many tests we've done in the US, unfortunately, it's probably not even 200,000 yet. So the good news here is there's plenty of supply of this product, and make sure that we can get it out there and offer it to as many people as possible. So again, like I said, significant regulatory approval. It was the the antibody test of choice for the Chinese CDC. What we show here on this slide is the approvals in terms of from the Chinese FDA. So again, it was the first colloidal gold test approved by the Chinese FDA. So again, hopefully you can see here, we've been very diligent and careful with what we wanted to work with to make sure that when we bring it here to the US, we've got some real provenance behind it, and we can feel very comfortable about the accuracy of the test. From an FDA and clear standpoint, as I mentioned earlier, we have a clear high complexity lab. That's our lab just outside Boston. We're covered by clear federally as well as the state of Massachusetts. So again, I think it's very important that when these tests are run, and although Brent quite accurately described them as pretty straightforward to run, you want to make sure this is done by a healthcare professional. And in our instance, we try to kind of have a kind of belt and braces approach. So not only is it being run by a healthcare technician in our clear high complexity lab, but it's also a product that is in with the FDA, already been approved by the Chinese FDA. It was submitted and we received an acknowledgement letter back from the FDA on the 22nd of March. So again, I think the turnaround time on the FDA at the moment is 2 or 3 weeks for these types of products. So we're anticipating that kind of any day now to make sure we've got that approval. To give you an idea though, our manufacturing partner also offers a real-time PCR product, and that's already been approved by the FDA. So again, gives me plenty of confidence in the team that we're working with, and also the accuracy and the level of quality that they have with the tests that they're offering, and that we're now going to offer here in the US. So again, you may be now asking, well, how do we get hold of the C19 kit? The first test that we're launching is the serum test, and that will be available via our website. You can see the link at the top of the slide there. We're looking to launch, as I said, the blood draw. We're validating a finger stick approach, which will be able to ship just a finger stick kit back to patients at home, and then they can ship that to the lab. The price for the test is going to be $100 plus $50 shipping if required overnight. For the serum test, that will be an overnight shipment back. It'll be an overnight shipment out to the patient or the provider, and then an overnight back. Again, the payment will be required before we ship the test kit out. And then the turnaround time from when we receive the test in our lab should be 24 to 48 hours. And we'll be providing that. There'll be an automatic upload to the provider's box account so that they can make sure box folder so they can see that happen. So some of you may be going, well, serum doesn't really work for me because I can't get to the, I don't want to open my office because, we're closing down for a period of time. So what we've thought of here is, rather than having to, you guys open your offices, is to offer at-home phlebotomy services. So we've signed up a small army of phlebotomists, in terms of 6,000 of them, and they operate all over the US. So we can cover pretty much every city in the United States. The prices for that mobile phlebotomy service are $80 for in-home collection. If you're a family of 3, then that just drops to $50 in total. Again, the idea of having this at home is because I know there's a worry that people shouldn't be going out, and there's lots of different orders in different states now about how we should be traveling, how we should be living our lives. So our concept was, let's give you the opportunity to remain safe in your home and have a phlebotomist come to the home. They can do it in the porch, they can do it in the garage. They don't necessarily have to come into the middle of your house if you're concerned about that. But again, this is a real good way of getting peace of mind to know are you negative or are you positive? And if you are positive, then you can then reflex on to the PCR test that Brent mentioned, which is the kind of the gold standard here in the US to make sure that you're a true positive. But it's as important to know, are you negative as well? And so that's why we're offering this service, to enable patients to be in the comfort of their home, and equally for physicians who are closed in terms of during this kind of trying time, they can talk to patients online. I know that there's a lot of telemedicine going on. We can drop ship to that patient's home, and then, as you can see here, if you then contact the email below, which is gina.gibo@avexiadiagnostics.com, provide her with a patient name, address, availability, and a phone number for the patient, then we can then set up that mobile phlebotomy service to be arranged for you at home as well. So in essence, although it's a serum test to start with, we wanted to go out with that because when you look at all the testing that was done in Wuhan, it was done on serum. So we want to take this test and use it in the way it was being used in, I guess, ground zero, as it were, in China. And so by offering this phlebotomy service, it gives you that peace of mind to be able to do that at home. And for the physicians who are closed at the moment and are working on telemedicine, it also gives them an ability of KBMO being able to drop ship and making sure we can get that phlebotomist service to the patient to give them peace of mind and get that testing continuing and happening. So that's really the end of the presentation. We want to thank you very much indeed for taking the time out to listen to us. As I say, I want to thank Dr. Dorval for his excellent explanation of the tests we've got, and also some background for you all on COVID-19.